I was thinking that this talk is a little bit like a description of a midlife crisis, but really an intellectual one rather than a marital and emotional one. So for many years I've worked on the nematode, Cinorhabditis elegans, as a model for studying the biology of aging. Not because I really care about why C. elegans ages, but because studying human aging it's a big challenge, given the complexity of aging and how intractable aging has been as a phenomenon to understand. So one strategy would be to say, well, if we could take an organism which ages, but which is very simple, and we could understand aging in that organism, that would give us a clue to understanding aging in an organism that we care about. So the strategy that seemed very exciting about 15, 20 years ago for trying to discover what aging is was to study mutants with altered aging rate. And this was a very exciting finding back in the early 90s, that one can actually obtain mutants where the aging rate is altered. And the idea was that if we could have long-lived mutants, we could then identify the gene that had been altered, and then we could clone it and sequence it, and that would lead us to an answer to this still unanswered question of what aging is. And what this led to was the discovery of a number of genes and pathways that control aging, some of whose effects on aging show evolutionary conservation. So the pathways control aging particularly in the C. elegans and Drosophila, the fruit fly, and the mouse. And this led to, as I think we've heard about earlier today, the discovery of the regulation of aging by growth hormone and the insulin IGF signaling pathway and the target rapamycin pathway. But the problem is knowing that these pathways control aging doesn't actually tell us what aging is, because these pathways are presumably working their effects by regulating some downstream proximal mechanisms, which in a way whose action really is working on the aging process itself. So I think really over the last decade in particular, there's been something of a kind of a block in attempts to try to identify what these proximal mechanisms are. It's turned out to be a major challenge. So it wasn't simply a matter of cloning the gene and finding out what it encodes. So what sort of processes might these be? And here I'm going to return to theory. And some of what I say is going to cover what we've already heard about, but much of it will, I think, be new. So there is an idea in the aging field, which one could perhaps call the standard aging paradigm, which is that organisms start off in a youthful state, and then through the accumulation of random or stochastic damage to the molecules of life, to DNA and protein and lipids and so forth, that leads them to become senescent. But the rate of the accumulation of this damage is not fixed. It depends on the efficiency of processes that protect against it, or if you like, the maintenance processes. And these would include things like detoxification, to remove the causes of damage, repair, to fix the damage, or turnover just to replace the damaged parts with brand gleaming new ones. So people trying to understand, so aging is the rate of aging reflects the balance between cellular maintenance and damage accumulation. So when people have been looking to try to understand how TOR and insulin IGF signaling and indeed dietary restriction are affecting aging, they tend to have in the back of their minds that it's probably something to do with maintenance and damage. And they tend to look for those sorts of processes in their analyses. Where does the damage come from? One idea is that perhaps it comes from oxidative damage from reactive oxygen species, such as superoxide produced by mitochondria. But the oxidative damage theory has been tested with increasing rigor in animal models such as C. elegans and Drosophila and the mouse, particularly through genetic interventions manipulating the antioxidant defense mechanisms. And then the results are mixed. Some of the studies support the theory and some of them don't, but I think over the years, the balance has been shifting towards more increasing studies, and particularly the more rigorous studies, failing to support the theory. So I don't want to go into a lot of detail here because this is sort of something that has accumulated over years. But the sorts of problem with the theory include the fact that treatments with antioxidants often don't slow aging, as the theory would predict, and that also overexpression of antioxidant enzymes often don't slow aging. Sometimes you can reduce the antioxidant defenses and you can increase the levels of molecular damage without affecting aging. And sometimes you can elevate the levels of oxidative stress and actually increase lifespan. So there have been a whole series of studies of this sort, which has led to a kind of crisis, really, I think, in this oxidative damage theory. I'd say particularly about 2009, people were really wondering whether the theory could perhaps be wrong. But this is a difficult, it's a difficult one because all of the work that I did or that I read about, you could never really say for certain whether the theory was wrong. What this work, as I would see it, what it does is it opens up, it sort of redirects the thought to some extent and opens up a number of possibilities. This has been presented already. So the question is the theory wrong? One idea is that it's still true, but that perhaps superoxide and hydrogen peroxide, which have tended to be the focus of tests of the theory, perhaps they're not so important, but perhaps other kinds of oxidative damage are contributing to aging. We heard earlier about glycation as a cause of damage, and that's a good illustration for this second notion, which is that the oxidative damage theory is a kind of half-truth. The real truth is that it's a much broader spectrum of molecular damage that causes aging than simply oxidative damage. And that's a sort of rather a commonsensical idea, and one that I guess I found most plausible up to a couple of years ago. But formally, there was always another possibility which one had to entertain, which was that what these negative results were really, what we should really learn from them, is that in fact molecular damage is not driving aging at all. That it's actually being driven by some other kind of process which leads to molecular damage. Because molecular damage does accumulate during aging, and it goes to very high levels during advanced aging. So I was sort of pondering about this quite a lot and discussing it with people. What else, what are the other alternatives? Because if you start to entertain this possibility that for all these years with this damage maintenance paradigm we've been barking up the wrong tree, the problem remains, well, if it's not damage, then what is it? What could be causing aging? This was a standard response. It has to be damage, because what else would it be? But then I was very struck when I came across this essay from somebody called Mikhail Vladimirovich Blagoslowny, who works at the Military Research Institute in New York. It's a Russian, called Aging Rosator. And this is an extraordinary essay. It's one of a series which presents a very mature, integrated theory of aging, which is very much derived from a reading of all of the recent work in the field and an attempt to deal with many problems and contradictions within the field. And I thought it was brilliant, this essay. So what what Blagosklony has done, it's rather like looking at a crossword where you're stuck with the crossword. And that's because one of the words in the crossword is wrong. And that stops you from being able to solve the crosswords. If you take that word out, replace it with the correct word, suddenly everything falls into place. And I think that's rather what Blagosklony has done. And what he has done is to remove from the crossword, if you like, the notion of the importance of stochastic molecular damage in aging. He takes that away and replaces it with a new concept called quasi-programmed hyperfunction. So what is this? Let me try and convey this. This is a very new idea to me. So, according to the old view, if you like, of aging, you have roughly two phases of the life history. You have development, which is programmed, a product of natural selection, essentially a nest of adaptations. You reach reproductive maturity, after which you have a non-programmed stochastic process of aging, the accumulation of damage. What the new theory says is that, that actually it's all a kind of program. That aging is not caused by damage, it's caused by a running on of these programs, but the running on is non-adaptive. So this is quasi-programmed aging. So it's controlled by genes, it's a function of processes that have evolved for the sort of the important part of the life history, but their running on is not optimized to keep the organism alive in the latter half of life. And the result is what he calls hyperfunction, which is an excess of activity, which can cause, it can give rise to hypertrophy or hyperplasia where you've got too much. It can lead to atrophy when you have too little. But in terms of what tends to limit lifespan, particularly, hypertrophy and hyperplasia leads to fatal diseases, such as cancer, such as cardiovascular disease, such as type 2 diabetes, and so on. So here's a sort of a potted version of his theory of what quasi-program means from Blagosklony. If you left water running after taking a bath, then a program for filling the bathtub would become a quasi-program to flood your apartment. So that's a simple version of it. Blagosklony also includes within his big picture the evolutionary theory. So, so one view of the relationship between the evolutionary theory and the damage theory was presented in this beautiful integrated theory, which is the disposable soma theory. So this would have it that somatic maintenance that protects against aging is costly in terms of energy and resources, and that creates an evolutionary tension between investing resources into maintenance or growth and reproduction. And so fitness can actually be increased by diverting resources away from somatic maintenance to growth and reproduction, resulting in a disposable soma. So this is a theory that integrates the two sides of the biology of aging, the mechanistic side and the evolutionary side. But of course, from the Blagosklony perspective, this part isn't true. And if this isn't true, then disposable soma isn't true. Blagosklony also points to a paradox that is thrown up by disposable soma, which is the effects of dietary restriction. So if aging is a reflection of partition of resources between somatic maintenance and growth and reproduction, then if you reduce the resources, you should reduce the available resources for somatic maintenance and shorten lifespan rather than increase lifespan. It's been suggested that organisms have evolved to increase somatic maintenance when food is low, which would imply that they decrease it when food is high. It's hard to see why that would happen. So that dietary restriction should shorten lifespan. So an alternative view to replace this is that nutrients are actually activating these nutrient sensing pathways leading to excess activity, photosynthesis, and pathology and aging. So if you reduce food levels, you reduce signaling, and you reduce uncontrolled biosynthesis, and you slow aging. So that's a simpler picture. There's no paradox here. So what this is arguing is that the soma is not so much disposable as bloated. So we can nickname that the bloated soma theory. Another long-standing question is, what is the nature of the genes that embody the antagonistic pleiotropy mechanism of the evolution of aging? And again, so we've heard about this theory before, and this, as I say, a long-standing history is what genes might embody this. And arguably, the insulin angiostigmine TOR pathway are a perfect candidate for this, for the genes underlying antagonistic pleiotropy. So what this would imply is that natural selection is optimizes your physiology to get you to reproduce successfully. And mutations that actually increase your growth rate, increase your reproductive success, increase your progeny production, may do so by elevating the levels of insulin angiostigmine and TOR. And in the long run, what that does is make you more prone to hyperplasia and hypertrophy, particularly in later life, and shortens your lifespan. So what I wanted to talk about briefly was work in my lab, where my lab's been testing the damage theory for about at least 10 years. And in the last couple of years, we started to test this hyperfunction theory. And it's been wonderful. It's a fresh breath of fresh air. So we've been looking at the worm through the prism of the hyperfunction theory. And this is work I'm going to show you is from a graduate student from Panama, a very industrious student called Yida. So one of the first things we did was we thought, well, if hyperfunction causes aging, C. elegans are very, very short-lived. They live only 2 to 3 weeks. So if hyperfunction causes aging, C. elegans should be full of it. So what we did was to look at the literature. There are hundreds and hundreds of papers on C. elegans aging, a tiny, tiny fraction of which actually look at the pathology of aging in the world. There's been very little interest in this. So we looked at those for any sign of hypertrophy during aging in the world. So here was one that we found right off. This is what happens in worms when they reproduce. So these are self-fertilizing hermaphrodites, the females that make some sperm for themselves. And they produce large quantities of yolk to provision their eggs. And it's made in the intestine. But then after 3 days, the worms run out of cell sperm, so they can't reproduce anymore. So the question is, what happens to yolk production after the end of reproduction? Do they switch the yolk production off? The answer is no. They actually continue to produce yolk. And the content of the worm of yolk goes up and up and up and up until the old worms are absolutely bloated with it. This is a young worm, and here the yolk's been tagged with green fluorescent protein. Here's a worm that's only 4 days old. You can see it's already full up with yolk. It's spread all over the animal. And this is, you can barely see this, but this is an electron micrograph through the body wall of the worm, through the intestine. And this dark brown stuff here is an old worm, which is just a solid mass of yolk. So that's hypertrophy. Within the gonad, in a young worm, you have a chain of oocytes maturing. But once the fertilization is finished, the oocytes continue to be made, and they just fill up the uterus and the ovary until they're completely packed like this. This is called stacking of oocytes. That's a form of hypertrophy. Later in life, these oocytes, they sit there for a while unfertilized, and then after a bit, it's as if they just say, oh, to hell with it. I'm sick of waiting to be fertilized. I'm just going to grow anyway. And they form these huge DNA-rich tumors in the worm that actually fill the entire center of the body of the worm. This is very reminiscent of aging-related tumors. For example, prostate cancer, rather similar. So that's hyperplasia. This is hard to see, but this is here. This is that white line there. You can just about see it. Is a neuron which has been labelled with green fluorescent protein in the worm. This is a young worm. And here's a detail of a neuron in an old worm, and you'll have to kind of imagine this here, but these are branches in the neurons. So the old neurons in the worms, they sprout neurites. So they grow wildly, which is an example of hypertrophy. Here's one of the craziest examples. This is the body wall of the worm in transverse section. So this is the body wall muscle here. And you can see that's the hypodermis, that black layer there. And then this grey layer on the top is the cuticle. It's like the skin of the worm. This is a young worm. Here's the old worm. Look at the cuticle. The cuticle has gone from this to this. So what? It turns out that when the worms actually develop, the cuticle is synthesized by the hypodermis. But when they reach adulthood, do they actually stop synthesizing cuticle? No, they don't. They just keep on producing cuticle until they get this absurd, thick cuticle. These are rather hard to see, but these are pictures essentially showing lipid droplets accumulating in old worms. So, in short, in a nutshell, are worms during aging, do they show signs of hypertrophy? They show extraordinarily extreme forms of hypertrophy, but nobody had ever paid any attention to it. And of course the reason why is because it has nothing to do with somatic maintenance and damage. So we've started to look at these individual pathologies of aging to try to see if they are intelligible in terms of this quasi-program idea. And I'm just going to end with one example of this. This is the gonad of the worm. It's a long reflex tube here. You can see oocytes here and spermatheca, and here's some fertilized embryos here. Now, when the worms get old, there's a very striking phenotype in this distal arm of the gonad. Basically, it falls to bits, it disintegrates. And this was observed, first of all, by the great Cynthia Kenyon in UCSF. And you can define different stages of pathology. So here's a young, healthy gonad. There's an oocyte here. You can see the big oocytes. These are the mitotic cells in the proximal in the distal arm of the gonad. So this we call the stage 1. These are 5-day-old worms. The stage 2, looking a little bit rough at the edges here. And you can see that the distal arm is starting to get thinner. Stage 3, this is a 7-day-old worm. You can see now it's in very poor shape. It's starting to erode quite severely. And you can see one of these tumor-like masses growing here in the uterus. At stage 4, it's actually disintegrated. It's fallen into bits. So what's the possible cause of this? Of course, based on the old paradigm, we'd say, well, it's molecular damage is probably causing this. So we said, okay, let's try and think about this in terms of the quasi-program theory. What would this tell us? You know, let's use this as a guide. So the quasi-program theory would say, well, maybe there's something that happens in normal gonad, young gonad, that makes the cells disappear. And it turns out there is. There's actually apoptosis, or cell death, which eliminates about half of the oogenic germ cells. And all this is about, this is just, in order to make one big oocyte, you have to take the cytoplasm from a number of smaller ones. That's all that is. So it's a bit similar to nurse cells in mammals. So, this is, yeah, that's what I just said. So we thought, well, maybe a simple explanation from Blagoslavny's theory is that the apoptosis just carries on until it completely destroys the organ. So there's a simple way to test that, which is genetically to block apoptosis using mutation in a gene called SED3, which is a caspase. This is how we present the data. So this is 1-day-old worms and 8-day-old worms, old worms. And these are the gonad classes here. See, so the class 1 are the healthy gonads. So here's a bunch of healthy gonads on day 1. And then on day 8 we have a range of different levels of deterioration, but the most common state is class 4. So here the gonad is disintegrated. So what happens if we block apoptosis? We basically have now no disintegrated gonads, and the majority are in stage 2, which are sort of wholly intact but looking a little bit a little bit shallow. So, in fact, the disintegration is apoptosis dependent. So then we have a natural experiment, which is that in males, in the gonad, there is no apoptosis, because they don't have to consolidate their germ cells to make one big one. They make lots of little ones, sperm. And in fact, it had previously, yes, so that would predict that in the male, you wouldn't get gonad disintegration. In fact, no one had ever looked in males. So we looked in males. So these are hermaphrodites. This is day 1, 8, 12 now. So you can see this increasing disintegration with age. And if you look in the males, there's no disintegration at all. And most of even the very old males have wholly intact gonads. So that supports this idea that apoptosis is driving this disintegration. And that's just a picture as a day 12 male with a lovely intact gonad. One other thing that we noticed looking at these worms is that sometimes they had these massive terminal oocytes. And you can see that here. This is a stage 3 worm with an atrophied gonad. And this is one oocyte. It's massive, huge. It's much bigger than a normal one. Very healthy looking they were too. And there's a picture showing the oocytes getting bigger with age. So these are the last oocytes that don't get fertilized. They grow bigger and bigger. So what we reasoned was that in the young worms, you have your program, which is for oocyte maturation, involving germinal apoptosis. But in the old worms, the quasi-programmed apoptosis causes not only gonad disintegration, but for that last oocyte, it also causes oocyte gigantism. Because there's one oocyte there, and all the oocytes, all the pre-oocyte cells are pushing their cytoplasm into this to produce this mega cell. So to get at that, we looked at the relationship between the level of disintegration and the frequency of gigantism in the oocytes, and found a positive correlation. And we also found that the SED3 mutation blocks the appearance of giant oocytes. So very recently, we've got some new data, which I haven't made slides for yet. We found that if we increase oxidative stress, that has no effect on gonad disintegration, consistent with, that it's a quasi-program and not damage that's causing the disintegration. We've shown that mutations that increase the level of apoptosis in the germline actually accelerate gonad deterioration. And we've also confirmed that the very old worms still have apoptosis going on, that there is quasi-programmed apoptosis going on. So what this I think shows very nicely is how a quasi-program can give rise both to atrophy in the form of gonad disintegration, but also hypertrophy in the form of oocyte gigantism. It also accounts for the sex specificity of the pathologies. So we have essentially an entire mechanism for a pathology of aging from beginning to end, without any reference to damage. And I think overall our view is that the hyperfunction theory provides a full and simple account for a major age-related pathology in C. elegans. So I think this is a very much a viable theory, and although possibly C. elegans is a particularly Blagoscloni-esque organism, in that its levels of hyperfunction, of hypertrophy and hyperplasia are very extreme. So I'd just like to end with a couple of comments. In 2001, it became clear that the growth hormone and IGF-1 signaling particularly was regulating aging. So we wanted to find find genes that control aging, find out what they encode, and that would lead us to aging. So we got to that point, and arguably, looking at it now, the identity of these genes was screaming the answer at us. Growth, what's causing aging? Growth hormone, insulin-like growth factor, TOR, which is a regulator of growth. Everything was telling us that, that growth is basically intimately linked to aging. But at the time, we argued our way out of it, and we argued that this must just be some sort of pleiotropy unlinked to aging. And why did we do that? Of course, because we were thinking in terms of the damage maintenance paradigm. And this is a rather extreme statement here. One thing that has crossed my mind is that when science moves from its sort of primordial state to a mature state, what often happens is the replacement of what's sometimes called the folk theories with actual scientific theories. So folk theories would be ideas like, for example, in classical physics, that heavy objects will fall faster than light objects, right? Or classically, that obviously the sun goes around the Earth, since you see it crossing the sky. In many ways, the damage theory has all the hallmarks of a classic folk theory, right? Things, old things, complex things, fall apart, they wear out. But I think there's a good chance that this hyperfunction theory and the broader framework of ideas may, in fact, be replacing our geocentric model with an heliocentric model and actually bringing aging into a sort of a new realm, new time. So just to thank the people that did a lot of the work, the Ross work and the hyperfunction work, and to thank Mikhail Vladimirovich, who's sort of from far away, but been a helpful advisor and supporter.